Hi everyone, my name is Hannah Jarvis, and this is my presentation on utilizing the UAS-Gal4 system of protein knockdown to observe the loss-of-function phenotype of P53 in Drosophila melanogaster. This is a project I started at the beginning of the semester in an advanced genetics course, and our goal was to knock down or reduce the expression of a GFP-tagged protein using RNA or protein knockdown to generate a novel phenotype. My protein of interest was P53, which is known as the guardian of the genome and also known for its role in the development of numerous cancers in humans. It is a tumor suppressor gene that encodes for a transcription factor to activate genes responsible for cell cycle checkpoints and stimulates apoptosis in response to genotoxic stress. We will be examining this P53 protein knockdown in Drosophila melanogaster, because it is a great organism to use as a model for their short lifespan and their short gestation period, which is a little over a week. And they're easily manipulated genetically due to their genetic simplicity, because they just have a set of X and Y sex chromosomes, as well as a second and third autosomal chromosomes. And they also have a small fourth chromosomal fragment, but it doesn't carry that much genetic material. And they also have evolutionarily conserved pathways that can be easily studied to provide insight to human diseases, such as the P53 mechanism. In order to observe P53 in ovaries and testes, I utilized the GFP and the UAS-Gal4 system. So GFP is a green fluorescent protein that identifies a protein of interest in tissues by being tagged to your desired protein of interest. So wherever there's a green fluorescence on the microscope, we know that our P53 protein is there. And the UAS-Gal4 system is a binary system that can be used as a method for gene knockdown to identify novel phenotypes due to a loss of function in a tissue-specific pattern. So the UAS component is an upstream activating sequence that can be tethered upstream of a gene sequence, and Gal4 is a transcriptional activator that binds to this UAS, and its expression can be controlled in a tissue-specific manner. The UAS is also linked to whichever molecule you want to knock down, so either RNA or protein, and the degrade FP is used to knock down protein, and it inactivates GFP-tagged proteins in a tissue-specific pattern, which is determined by the Gal4 factor. This contributes to the overall understanding of gene function in an organism. The UAS and Gal4 elements are usually kept separate during fly crosses until the last cross, and once they're combined, the Gal4 protein will activate the UAS element to express its associated degrade FP, which physically guides the GFP-tagged proteins to the proteasome for degradation, and this will lead to our loss-of-function phenotype. And this picture shows that the final cross of the UAS degrade in the Gal4 line allows the Gal4 to activate the UAS, and the degrade FP is produced so that it can degrade the GFP-tagged protein. So in this experiment, the UAS-Gal4 system was used to knock down the expression of P53 in the germline of Drosophila. So the Gal4 component for ovaries is nanos, and the Gal4 component for testes is T100. And we know that for ovaries, there are nurse cells that surround the developing oocyte, and they synthesize proteins and maternal mRNA to be deposited into the developing oocyte. And these nurse cells must undergo apoptosis to generate a fertilizable egg that can support a viable embryo. We can see this in this picture right here. These pink colored cells are the nurse cells. And in the last stage of development for the egg, this mature egg doesn't have any nurse cells, and we know that they need to undergo apoptosis. So if there's a knockdown or reduced function of P53, I think the defect could prevent the apoptosis of nurse cells and cause sterile phenotype. And we also know that p53 contributes to spermatogonia maturation. And so knockdown could also disrupt spermatogenesis and cause a sterile phenotype in males as well. So this study will observe the biological effects of knocking down p53 in the germline of Drosophila melanogaster. So onto my methods. Flies were cultured in vials containing Carolina blue fly food, and they were incubated for most of their lives at 28 degrees Celsius. This, at warmer temperatures, their life cycle is sped up, and we're able to get our desired offspring faster. But when selecting for virgins, they were incubated at 20 degrees Celsius so that it could slow down their life cycle, and virgins would not mate with males. I'll talk about why that's important a little bit later. Our fly stocks were obtained from the Bloomington Drosophila Stock Center, and they were initially mated with a multiply balanced stock of markers and balancers. And the balancers are very important because recombination of these results in a lethal phenotype. So there's one for the second chromosome and one for the third chromosome. Curlio, or CYO, is the balancer that prevents recombination for the second chromosome, and its phenotype is curled wings. And TM2 is the balancer for the third chromosome, and its phenotype is small bristles on the halteres. And then the multiply balanced stock also has markers, and these were used to follow our crosses. We can use the phenotypes of these to follow the genetic elements to ensure that our crosses were successful. So the scutoid marker is on the second chromosome, and we used this to ensure that the chromosome genetic elements of the UAS degrade and the UAS gal4 were within the phenotype. So we selected against scutoid, which is the loss of the back4 scutellar bristles. We use those to ensure that the degrade and the UAS were there. And then to ensure that the third chromosome p53 GFP was in the genotype, stubble was selected against, which is when the scutellar bristles are shorter than usual and they don't cross. We didn't want to have those in our crosses, or else we wouldn't know if we actually were getting our desired offspring. And collecting virgins is also really important to get the desired offspring that we want, because female Drosophila only remain virgins for 8 to 10 hours after enclosure, which is when they're officially adult flies. And they can actually store sperm after one single mating, so they have to be collected within this time. If they store this sperm, then they can mix the genetic material of multiple fathers, and so then we wouldn't know if we had our desired offspring. And also, since p53 is homozygous lethal, we also obtained a mutant p53 strain, which contains a deletion, so that in our final fly, we don't have homozygous lethality. And then we also dissected ovaries and testes from the adult flies of the original p53 stock, as well as a UAS RFP cross. So this UAS RFP confirms the expression of the gal4 driver in our specific tissue. And the adults were dissected in PDS, and they were fixed in a 4% formaldehyde and PBS solution, and they were also stained with DAPI. DAPI provides a staining to the nuclei of tissue, and this can be used to compare to the staining of the green fluorescent protein, which is where our p53 tagged, or p53 GFP protein is. So we can compare the GFP staining to the DAPI staining and see where p53 is expressed. So here is my experimental cross scheme. This was the proposed cross scheme of the entire project for protein knockdown in nurse cells of Drosophila. And the crosses A, B, C, and D are for the degrade element, the mutant GFP P53, and then the normal wild type P53 GFP, and then as well as the Cal4 driver. So here you can see we selected against scutoid for these offspring and against double for the P53 offspring, and then also again selected against scutoid for the Cal4 driver. Towards the end of, or towards a couple weeks ago, the NOS Gal4 and the RFP cross was not producing any offspring. So we weren't going to be able to validate that my driver had expression in the ovaries. So I switched to testes. So this cross scheme would be the same, except for this Gal4 driver would be T100 for the testes. And finally, a couple days ago, we had some flies pop out for that. So we do have a validated expression of the nanoscal4 driver in ovaries. So moving on to my dissection results. Here, this top set of images were taken on the A1 confocal microscopy. And the first image is the GFP expression. So this is all the green staining is showing where P53 is. And we can kind of see that it surrounds the nuclei within the egg chambers, the developing egg chambers. These right here are the dorsal appendages of a mature egg. So the mature egg is kind of in this top corner here. And then the second photo is the DAPI staining, which is for the nuclei of cells. So it shows our cell density. And then C is an overlay of the two. So you can kind of see how the green fluorescence surrounds the blue DAPI staining of the nuclei. So from these images, we can see that P53 seems to be paranuclear or in the cytoplasm. In the center of the images, yeah, they show the developing egg chambers. This one is in earlier stages and this one's in later stages. And these tissues were dissected from the P53 GFP study. Then these images are the offspring of the UAS RFP, which is a cherry staining, this red staining, and the NOS-Gal4 driver line to confirm that my nanoscal4 driver is showing expression in the ovaries. So again, we have our DAPI staining, which stains the nuclei, and then we have the NOS-Gal4 driver, which is stained with the cherry, which also seems to be around the surrounding the nuclei. So we know that because we validated our driver, we know that when we do the final cross with the UAS degrade and the Gal4 driver, we know that P53 will be degraded in the ovaries. So for my conclusion, we did have a couple difficulties throughout this experiment. Earlier in the semester, a lot of our fly cultures were subject to bacterial infection and many of our fly crosses died. So we had a fly antibiotic that we put on a chem wipe and put inside the vials. And this helped a lot with having our crosses survive and generate offspring. And I mentioned earlier that the NOS-Gal4 with the RFP was not producing offspring. So in the meantime, I was trying to observe testes, but our cross was successful, which is why we have these images here. And then with the fly stock and crossing the fly lines for the future, I would like to continue with the experimental cross scheme I showed before and to get the final desired offspring to create a balanced stock of the P53 that is knocked down in the ovaries as well as testes. And I can do some further analysis on the new phenotype, the loss of function of P53. And the conclusion I came to with the imaging is that P53 protein expression in the ovaries, the GFP signal shows that the cytoplasm and, that P53 resides in the cytoplasm as well as paranuclear. and it has a more intense signal around the large nuclei observed by the Daffy staining. So with this data, my hypothesis remains that the knockdown of p53 protein will disrupt the maturation of oocytes because apoptosis of nerve cells will be inhibited or reduced, and as well as the disruption of the maturation of spermatocytes. So I predict that both of these knockdown processes will lead to a sterile phenotype in both males and females of Drosophila melanogaster. Thank you so much for listening to my presentation. Let me know if you have any questions and I'll be happy to answer them. Thank you.